Dr. Kristin Bilyeu is a research molecular biologist with the USDA ARS Plant Genetics Research Unit and an adjunct professor in the Division of Plant Sciences at the University of Missouri. Her research focus has been to increase the value of soybean by utilizing variant alleles of genes to improve seed composition and ultimately plant performance. Her expertise is in the translation of basic research to applied challenges utilizing biochemistry, molecular biology, genetics, and genomics to increase the efficiency of plant breeding and development of improved soybean varieties. Dr. Bilyeu has an active field and laboratory research program that utilizes natural and induced genetic variation to determine the optimum allele combinations to improve soybean traits that provide value to crop production and nutritional enhancements to our food supply in the US and developing countries. And on a personal note, Kristin Bilyeu is my PhD research advisor, and I feel like I am one of the luckiest students that I know because I had such an amazing advisor. Not to be too emotional, but I am really excited to be in this position now and be able to invite Kristin to give this keynote speech.
Thank you, Carrie. Is everything good? My screen?
Perfect. Everything. I can hear you just fine.
Okay, good. Thank you, everyone. Welcome, I guess, to North Dakota. It's kind of interesting to be in North Dakota and to be sitting in my home office where I've been for the last year. So if anyone else is feeling the Zoom pain, I'm right with you. And so today what I'd like to do is tell you about something exciting that's going on in my research life. So this isn't going to be a collection of the research that I've done in my career. It's actually something relatively recent that I've gotten into over the past, 5 or 6 years, since 2015, really, when I went to a workshop about Genome Wide Association Studies. So I want to talk to you today about what I consider applied soybean genomics. And I made up applied genomics. I'm not sure it's a thing, but it is in my mind. And I want to explain a little bit about what that means to me. And I hope to you through the next 40 or 45 minutes or so. So to me, genomics is really the study of genes in genomes and utilizing genome sequence information in a powerful way. And that powerful way is to improve crops. And whole genome sequence or genomics science is really it applies to every living thing because everything has a genome. And so we can characterize that genome through sequence analysis. And so it's really a unifying field in biology. But because most genomes are quite large in terms of data points, it's a big data science. And unlike some of the prior research that I did, I found that doing work in genomics requires some fluency in bioinformatics and the development of tools to deal with the data. That isn't really how I was trained. And so it's fun for me because I have an opportunity to learn new things and to explore. And I think it's really an empowering field as well. So how do you apply genomics? Well, how can sequence data solve problems? And I'm so glad that I followed up with Aaron Lorenz's presentation that was just over a few moments ago, because he talked about how he's moving his breeding program to really incorporate this idea of breeding by design. And so taking classical breeding methods and modernizing them by taking information about the genome, and especially incorporating our new understanding of genes that cause phenotypes into those models or into parent selection. And so to me, that's what applied genomics is. It's taking genome sequence information and big data science and applying it to improving crops. And wouldn't it be great if there were giant arrows pointing us to places in the genome where genes resided that had variants that caused phenotypes that were interesting or important to us? So this is sort of the perfect world. And it also describes the soybean genome that's really 20 molecules. It's 20 chromosomes in a haploid state. And the whole soybean genome is about the third the size of the human genome. It's about 1 billion base pairs in length. And it's organized in these chromosomes, and that presents us with an opportunity through breeding and also through research to understand all of the genes that are present in the genome. And so we have probably 50 to 60,000 more or less functional genes in the soybean genome that are organized all along these chromosomes. So in the neighborhood of about 2,500 genes per chromosome that control the biology of soybean, and really the biology of all living things is held in its genome. And so I'm going to come back to this at the end because it's kind of a fun thing for me to think that in the perfect world, we would have assistance in finding the genes and the variations that control the phenotypes. And that's what I'm going to talk to you about today. And what I'm going to focus on is an association analysis that we call GWAS, for Genome Wide Association Studies. And this is a genetic technique that allows you to take a panel of what I'm calling genetically related samples, but not siblings, sort of more distantly related samples, and use some genome sequence information from those individuals and some statistics to connect places in the genome that have genes that control phenotypes that you can measure. So we'll just call this GWAS, and Aaron also showed some GWAS results in his presentation, so I was happy to see that because it's something that's relevant, and Carrie mentioned in some questions that she had to one of the former speakers, a GWAS paper. So I think that GWAS is relevant today. And one of the things I wanted to point out is that my view of GWAS is that usually what happens is we get this sort of pixelated, low resolution view of what's going on. And there's an opportunity to sharpen up that image and supplement it with high density information, with big data information, so that we get the real picture of what's going on. And so this is, of course, just the Mona Lisa in a pixelated and full digital reproduction way here. So this is really my goal, go from a low resolution to the exact picture that we want to see. I also want to bring up that GWAS differs a little bit from classical genetic mapping studies. So Aaron also mentioned some genetic mapping that he was doing. And this is a sort of the classical genetic technique where a population is made with two parents, and the objective of the experiment is to identify a region in the genome that contains a gene that controls the phenotype that you're measuring. So this is genetic mapping, and what distinguishes it from this association mapping is that genetic mapping provides the researcher with boundaries. So if you're looking for something on this map, it's a little place in Houston, Texas, near where I used to live. Genetic mapping result would be that what you're looking for is within this box with the red boundaries. No matter what, it's not out here, it's within the box somewhere. That's a genetic mapping result. And the idea is to make this box smaller and smaller and smaller until there aren't very many candidates within the boundaries so that you can identify the genes. I think Aaron said he was down to 17 genes in his genetic mapping experiment. But he also mentioned some GWAS studies. And as I said, that's a little bit different. GWAS also points you to a location. So you might get on the map a dot, but that dot doesn't mean what you're looking for is exactly there. It just means it's in the neighborhood. So what you're looking for might be here, or it might be here, or it might be here, it might even be over here. The GWAS results point you to a location, but there are no boundaries. So I just wanted to point that out because we'll be talking about GWAS here in a little bit. Okay, I guess about 3 years ago, a group was funded that I co-lead with Trupti Joshi at the University of Missouri to do an applied genomics project with soybean. And the first thing that we did was we took a while to develop what we call the Soy 775 data set. And I'm going to take this slide to explain what that is. The Soy 775 data concept is that it's a set of genomic positions within the soybean genome and a set of soybean accessions that we have complete data for a big data set. So all of these 775 accessions of soybean have been whole genome resequenced and mapped back to version 2 of our Williams 82 reference genome. And if you'll remember, I said that the soybean genome has about 1 billion base pairs. And so the data generation is quite tremendous, actually, for these 775 whole genome resequenced accessions. They come from these different published research sets. So as a group, we didn't conduct any of the sequencing ourselves. We just did analysis. So there were 775 unique resequenced accessions from these different projects, which had different coverage levels. So this is a 11x coverage of 293 samples. This one was an average of 15x coverage on 302 samples, and so on for these different sets. So there were 775 accessions. They were resequenced to these different pretty good depths, and we curated all of the single nucleotide polymorphism, and which are SNPs, and small insertion or deletion positions within this set of 775 accessions, and curated those. And in the end, we had about 36 million variant positions. So out of the 1 billion bases of soybean DNA, we found that at least one of our 775 accessions had a variant from the reference for 35.7 million positions. And then for each of those positions, we use a tool that's called SNPF, that's available online, to determine whether or not each of those variant positions and what the variation was. So if it was a SNP, it might have been a change in the reference, it was a G and a C in the alternate case. Or if it was an indel, maybe it was 3 bases in the reference and 1 base in the alternate case. And whether those variant positions were in the annotation file of where the soybean genes lived within the genome to predict an effect. So SNPF is a variant annotation and effect prediction tool, and it allows us to give more information to our curated positions. So whether or not the position resides in a gene, and if it's in a gene, if that alteration has a predicted change in the sequence of that gene, essentially. The other thing I want to point out about this Soy 775 data is that it consists of a variety of types of soybeans. So the total is 775, but we had 111 wild soybean accessions, 190 cultivars, and 474, or the majority of the accessions, were land races. And I think about 600 of these accessions are present as plant introduction accessions that are available in our GRIN repository. So most of the accessions can be ordered from the GRIN and have some advantages as being held in the GRIN. So this is our Soy 775 data set. We spent quite a while developing this data, curating the information, managing the data because it is big data. And then what I'm going to tell you about in a little bit is how we're using this data. And I'm going to take you in kind of a weird circle today, I hope, if we all stay together. And what I hope to do is start with something known, take you through my thought process for a tool and I'm hoping a revolution in an analysis technique that empowers other researchers. And then we'll come sort of full circle back to something that's unknown and use the tool. And hopefully I'll convince you that this is something that's reasonable for researchers to use, especially in soybean, but also in other crop species as well. So the thing that's known that we're going to start with is the example that I'm giving here of a soybean gene. And this is some screen information taken from the Phytosome website. This is a repository of data. And this is an example of the gene, which is listed with these dark green boxes as the coding sequence interrupted by the intron sequence. And then all these little blue lines are variant positions that have been cataloged at the JGI site for this gene. And this is the tawny locus gene. It is responsible for the colors of the hair on the pod and the stem. So that's either tawny or gray. Pubescence is the name of that hair. We have a naming system for soybean genes. And so this particular gene in version 2 is named this. And then there's a biochemical name for the gene that we abbreviate F3'H. And it's listed here along with some more biochemical information. We know the identity of this gene for the tawny locus because of some work done previously by Lila Bodkin's group. And we know the exact position in the genome that makes this gene either the tawny version or the gray version. The tawny is the functional version. And then there's a frame shift mutation at this very position on chromosome 6, 18 million and some, that in the reference in Williams 82, it has a T and a C at that position. But in the alternate, it has just the T. The C is missing. So we call that a frame shift. And this is what we will call the causative mutation, or CM, for the T locus, and I'll use this throughout the talk. So this gene is one of many cloned soybean genes, and we're going to use this as an example. So I want to talk about what a typical soybean GWAS looks like. Aaron Lorenz did show a couple of GWAS results in his presentation, and like I said, Carrie mentioned one, and I will use this example repeatedly of the 3 parts of a GWAS, and they're listed here in these blocks. So the 3 parts of a GWAS is a panel of accessions. In this case, there were about 4,800 accessions from the GRIN. These are soybean accessions. This is our panel. And for that panel, we have 2 things. We have a genotype, or markers. In this case, we used the soy SNP 50K markers that are part of an Illumina product that is available for almost all of the soybean GRIN accessions. And we consider this a low density marker, even though there are 50,000-ish markers in the set. The density is actually quite low if you consider the variation that's possible in the soybean genome is probably around 40 million positions. So this is just a small percentage of those, maybe 0.1% of the total variation. So you have a genotype, and the other part that goes into GWAS is you have a phenotype for each of these accessions. And in this case, we used hilum color. This was either black or gray hilum color. And in Carrie's talk, she talked about the importance of hilum color. And there are several known genes responsible for that. Some of them have been cloned. So if you have these 3 parts, a panel of accessions, a genotype, and a phenotype file, you can generate an association, which is a statistical test for each of these marker positions in the soybean genome, and you get a Manhattan plot. And the output looks like this of a Manhattan plot. So on the x-axis are the 20 soybean chromosomes, and each one is color-coded a little bit differently here. And what the colors are made up of are the individual markers, along with, on the y-axis, their statistical significance in the, and we can think of it as their confidence level that they are associated with the phenotype. So it's like a t-test for every marker, asking the question, how much confidence do we have that there's an association between that particular marker and the phenotype? And where that confidence is low, the negative log p-value is low, and where that confidence is high, the negative log p-value is high, and often it forms this peak, which is supposed to look like a Manhattan skyline, depending on the results. So in this case, for hilum color, black or gray, there were 2 peaks, one on chromosome 8 and one on chromosome 6. And the highest peak is a special one. We'll talk about that later. But this marker then has a position on chromosome 6, for example, that we know is near the tawny locus that I just described. And this marker is near a locus that we know to be the inhibitor locus. And that makes sense for what we know about this. So what happens when the typical researcher does a soybean GWAS or any kind of GWAS and they get a result? Generally, they look at the location of the significant markers and they take a guess for what might be around there that is likely to be responsible. So if you're using a phenotype that a lot is known genetically about that guessing can be pretty powerful. But many times you're developing Manhattan plots and these peaks on different regions where you have no idea what gene is responsible. And so for the most part, when people do GWAS, they don't get to the gene that causes the trait. So they might say, well, this peak is near something that I know, so I think it might be the tawny locus, but that's pretty unsatisfying, especially as a molecular biologist who likes to get to the bottom of the mystery for what is actually connecting the phenotype and the genotype. So because for many reasons, and one of them that statistics is not really my strong suit, I had a different way of thinking about this association between the phenotype and the genotype. And that's a pretty simple way of thinking of it. And that's a term we call accuracy. Accuracy came up earlier today as well. And it's a mathematical concept that most people understand. And I'm using it here to describe the direct correspondence between two things, and that would be the phenotype and the genotype. So I've kind of thrown to the side the statistical confidence method, and I'm looking at direct correspondence. I'd just like to go through what I mean by that. Again, for the tawny locus, remember this is cloned, and here's an image from the cloning paper where the hairs on the pod in this left side example are brownish or tawny, and the hairs on the right side pods are white or gray. So this is tawny and this is gray. And we know the gene, we know the exact location of the causative mutation. So in order to explain accuracy, I just made a little schematic here. So 100% of the accessions that have tawny pubescence have the reference base at this position for the causative mutation. And because that causes the phenotype, absolutely none of the tawny accessions have this alternate base, which happens to be an indel at this position. So you have 100% accuracy for the reference and 0% for the indel for tawny. And then similarly, for all of the accessions that have gray pubescence, they all have the indel, the causative mutation for this phenotype, and none of them have the reference base. Otherwise, they would be tawny. So this is accuracy, and there's two parts, and we call the parts of accuracy that is identical to the reference wild type accuracy, and the part that's identical to the alternate, the mutant accuracy. And when I say average accuracy, what I mean is the combination or just the average of the wild type and mutant accuracy. So for most of the talk, I'll just say accuracy, but I'm talking about this average accuracy that takes into account the direct correspondence of a phenotype with a genotype, with 100% being completely accurate and 0% being not accurate at all. So this idea of accuracy became such an interesting and intriguing thing to us that we built it onto a tool an existing tool that we were using, and then realized that it was so important, it needed its own tool. So we developed what we call the Accu Tool, and it's available on soykb.org. If you go there and there's a tools drop down, you can find Accu Tool listed and go to the page directly that will take you here. And the Accu Tool calculates accuracy for you for much like a GWAS, a panel of accessions that has genotype and phenotype information. So it uses an input interface where you select the chromosome and the region that you want to look at, and you provide a phenotype file, and then it does all of the work, and I'm going to go through that. But first, I want to say that the Accu Tool has under its hood the 35.7 million genotype positions of SNPs and indels from the Soy 775 accession panel, and it has those 775 accessions. So it's a matrix that is, well, we separated it by chromosome, but in general, it's for every one of the 35.7 million positions for every accession, whether it has the reference or the alternate base there. And then for each of those positions, as I said before, we know if that position modifies a gene. We also have highlighted within this data the soy SNP 50K positions, because that helps us link things together. And these accessions, as I mentioned before, are a combination of wild soybeans, cultivars, and land races. So that's what's under the hood of the Accu Tool. And I had previously mentioned that GWAS is composed of these three things, right? Well, the Accu Tool is also composed of those three things, and some of them are fixed. So the panel of soybean accessions is the Soy 775 panel. It's fixed, so it's under the hood, it doesn't change. The genotype are the variants, so you have a possibility of the 35.7 million variant positions, but you sub-select for a region that you're interested in. And this, I'm going to say, is sort of semi-fixed. And then the phenotype is something that you provide. So in this example, which I'm going to use as a forward example, I put in for the 775 accessions where I knew the phenotype data for either tawny or gray pubescence. You upload a file that follows a template, and you click on a few things, you select your region, and then you ask the tool to calculate for you the accuracy against all of the other positions in the tool. And the output looks a little bit like this. So these are the results that you get. Remember, the three parts of association analysis are the panel, the genotypes, and the phenotypes, and I have those listed here. And the results are a table that you can sort. So I cut this off here, but this is chromosome. We're talking about chromosome 6. It has the variant position listed, then the average accuracy that I described. Some other things I'm not going to go into right now, the gene and the effect. So the effect is the SNP-F effect that I mentioned before, whether it's in a gene or not. And it allows you to sort by any of these columns. You just click on the little gray arrow. So once for ascending and twice for descending. So in this case, we put in a phenotype of pubescence color on chromosome 6 in a certain interval for all of the variants. And we ask it to give us the accuracy for those. And I sorted in descending order. So here's the highest accuracy position. And if you want to connect the phenotype to the genotype, then you look for high accuracy positions within any genes here. And actually all of these top high accuracy positions are in the same gene. And the second position is the frame shift variant that causes the tawny or gray phenotype, and that is the correct position for that. So association is a little bit like GWAS, but it's a direct correspondence. And it doesn't take into account population structure or anything like that. It's just a direct correspondence. But at the same time, I hope you can see that with just a little bit of information, it's extremely powerful at identifying causative mutations or candidate genes at the very least. Okay. So now I want to go around my circle a little further, and we're kind of going to go backwards for a little while. So we use the Accu Tool to do something that we call inverse GWAS. And that is, instead of our normal three parts of a GWAS, as our panel has our variant positions, for the phenotype, instead of it being a real phenotype, we've substituted a special phenotype, which is the causative mutation for the tawny gene. So remember, I told you that causative mutation is on chromosome 6 at this position. And so previously we put in a phenotype file here that was a list of real phenotypes, tawny or gray pubescence. But the tool allows you to put in a position in the genome as a phenotype. So we put on chromosome 6, this is the position of the causative mutation. And we're now asking the question, what other variants have high accuracy to the causative mutation of the tawny gene? And the result, when you ask the tool to calculate that, is not that surprising. The causative mutation has 100% accuracy to the causative mutation. So again, if you sort by descending average accuracy, the tool returns with complete accuracy the position that you put in, the causative mutation. But kind of interestingly, it shows high accuracy see high accuracy numbers here, with other SNPs in the same gene. And actually, this is the same list of positions that we got when we used the real phenotype as the input. So direct correspondence through accuracy is highlighting, I was going to try not to say it, but the linkage disequilibrium that exists in the genome. And so this might not be all that interesting on its own, but there's a special way we can utilize this. And that is, as I mentioned, we'll go back to the input part of the tool. We put in the causative mutation for the tawny gene. And this is why we call it inverse, is we're going backwards here. We're asking if we know the causative mutation and we want to know what SNP 50K positions are highly accurate with that causative mutation, we just select instead of looking at all positions that are high accuracy, we're going to say, look at only those that are soy SNP 50K positions. So we've changed the genotype file here by filtering for those that are only the soy SNP 50K. So this is still part of inverse GWAS. And the result then, when the tool calculates accuracy, is, again, we're sorting by descending average accuracy, is a set of the soy SNP 50K markers. They all start with this SS715 identifier. And sometimes the markers are within genes, and sometimes they have, they're intergenic. Sometimes they have gene effects. You never know with this marker set, but it just lists that information here. It's not particularly relevant. But if you use the causative mutation as your phenotype and you select your genotypes are 50K markers in this panel of Soy 775, what you can find is a list of very highly accurate markers to, in this case, the tawny gene causative mutation. And that might not sound interesting to you, but to me, this is, if something is 96.2% accurate for the status of the causative mutation for tawny, then it becomes a proxy marker for the tawny causative mutation. And what I mean by proxy is if you know the status of this marker, but you don't know the status of the tawny gene itself, you can, with very high accuracy, predict what the tawny allele status will be. And so we call that a proxy marker because it can substitute for the real thing. And I mentioned before that most of the GRIN plant introduction accessions have full 50K marker data available for all of the accessions. So if you wanted to know the tawny allele status for any of the soybean accessions in the GRIN, you could look at this particular marker status and predict it with very high accuracy. Actually, you can use really any of these very highly accurate ones. So that's inverse GWAS. And what happens then is that we can link what I called forward GWAS and inverse GWAS, especially for the tawny gene. So we'll go back to the first GWAS that I showed you that had hilum color, either black or gray, the 50K SNP markers, and now 4,000 accessions that we looked at. And we said we got a highly significant associated marker. This is a 50K marker near the tawny locus, and then these two others that are a little bit lower. Well, it turns out that this highly associated marker is the number 3 proxy SNP when you use the tawny locus as the causative mutation in an inverse GWAS. So we call that a synthetic phenotype. And actually, these the second and third highest associated SNPs in the forward GWAS were the second and the first proxy SNPs when we use the tawny locus causative mutation in the Accu Tool. So this is one of the ways I can link accuracy with GWAS, and I can now say with confidence that this is the tawny gene that is showing up in my GWAS that's saying it plays a role in whether or not the hilum color is black or gray. And because the Accu Tool is available, anyone can put their cloned gene in and develop some proxy markers for their favorite gene to try to see if what they're looking at is likely to be a cloned gene. And so we've collected a list of about 20 clone genes that occur with high enough frequency that we can do this in our Soy 775 data in the Accu Tool. And this is just a kind of a snapshot of some of those. We talked about the pubescence color tawny gene. This is the causative mutation position on chromosome 6. And I've included, I think, 5 markers in each case, 550K markers with the highest accuracy from the causative mutation. But I wanted to point out that one of the things that we find when we do this for different genes, this is E1 and light tawny and flower color and pod shatter and so on, is that for the tawny locus, the proxy SNPs are pretty far away. So 400,000 away, almost 300,000, 232,000 bases away from the causative mutation, very high accuracy. And sometimes they're very close. For flower color, there's a over 96% average accuracy that's only 9,000 base pairs away. For the pod shatter gene, there's a very, very highly accurate marker that's only about 4,000 bases away, almost in the gene itself, a 50K marker. And so proxy markers give you some power to analyze your GWAS results for a set of known genes, or when you have a candidate gene, you can look at the connection between a proxy marker and what's going on in your GWAS. Okay, so now I want to talk about that I've linked GWAS to INVERSE GWAS and the Accu Tool and accuracy as a concept. We have a larger strategy that we call GWAS to genes that is an analysis method to help empower people to discover the genes that are connecting their genotypes to their phenotypes. And it's meant to be used after GWAS. So this forward GWAS that I showed you all along, we kind of call GWAS 1. It's the first step. And I'm going to give you just sort of some slides and discussion about conceptualizing our GWAS to genes pipeline without going into all of the details. And so I connected the GWAS result with an inverse GWAS and a proxy SNP. And so if you don't know what your gene is, if you don't have the big arrow telling you this is your gene and after a GWAS, then if you get a highly associated SNP, we call that SNP a tagging SNP, and we're going to use that feature in the Accu Tool. So let's move on to a different phenotype. This one is leaf type, and it's either ovate leaflet or lanceolate leaflet shape. And if you do a GWAS on your custom panel of accessions, these are GRIN accessions that had 50K genotype data. So again, the 3 parts of GWAS, your panel of accessions for which you have genotype and phenotype data. We can do a GWAS with 50K and leaf type, and the result is this Manhattan plot. It looks, again, although it's in black and white or black and gray, the 20 chromosomes, each marker, and the confidence that that marker is associated. And so we get a marker here on chromosome 20 that has this identity that we happen to know is near the lanceolate locus or LN locus. And so the marker itself from the GWAS has a position. And so I told you before that we could use a causative mutation as a phenotype instead of a real phenotype. Well, we can also use a tagging SNP as a phenotype because it's just a position, just like a causative mutation is a position. So we take this tagging SNP marker, its position, it's on chromosome 20 at this location, and we feed it into the Accu Tool. Again, we call this, when it's not a real phenotype, we call it a synthetic phenotype. So the 3 parts of the Accu Tool, it's always the panel of Soy 775 accessions. We have those 35.7 million variants to choose from. So we narrow it down on chromosome 20 to some interval. And then instead of a real phenotype, we're using the tagging SNP as a phenotype. And that shows up in the input down here. So previously we used a real phenotype. We used the causative mutation for the tawny gene in one case here. But this time we're going to use the tagging SNP. That's the result of GWAS 1. And in the prior analysis, I showed you how we could highlight the soy SNP 50K positions only. Well, we don't want to do that here because this is a 50K position. But instead, we want to highlight those variants in the genome that cause changes in genes. So you click yes here to the amino acid modifying variants. And the results, again, come up in this table that have chromosome position. This time, we're using a different format of accuracy. This is what we call the pessimistic accuracy. That's kind of the worst case scenario. So every time there's missing data, it counts it as wrong. We've sorted in descending order again. And I've highlighted this column with effect. And luckily, I can use PowerPoint to add a nice flashing arrow here. So the fourth highest pessimistic accuracy position result is in this gene, glyma.20 and G116200. There's a missense variant in this at this position, and it causes a D at amino acid 9 to histidine change. And this happens to be the causative mutation for the cloned gene for the LN locus. So by using the synthetic phenotype from the tagging SNP, we were able to get a short list of candidate genes using the Accu Tool, and it turned out that the fourth most accurate one to the tagging SNP was the causative mutation for this gene that's already been cloned. And we've done this for many genes that have been cloned, and I would like to say that each gene is unique, has a unique set of circumstances, but it's kind of remarkable how good accuracy correlates with the causative mutations in genes. So finally, I want to follow up by a gene that's not cloned and tell you a little bit of a story there without going into all of the details. So Carrie mentioned, and also Aaron mentioned, seed coat color is important for soybean. There's a classical locus that distinguishes brown or red brown seed coat and hilum color. And I have a little image of those colors here. I know it's really hard to see, but brown doesn't really have any red in it, and red brown does. We know some of the genetic loci responsible for that, the combination. And so we did a forward type of GWAS or GWAS1. The three parts of that were a panel of custom GRIN accessions, 50K data for the markers, and then red brown seed coats or hilum and others. And this particular GWAS included yellow seed coat colors as well. And so we got peaks on different chromosomes. On chromosome 8, we know that the I locus resides there and the O locus. So if we look at the inset, this is a zoomed in region of this data on chromosome 8, where the I locus has a peak, the O locus has a peak. And then this is even zoomed in further. And these are the results of this very kind of stunted looking peak here, where there are several 50K SNPs that are with some confidence associated with the O locus, but they aren't really outstanding negative log p values. And it turns out that none of these is particularly great at getting a list of nice candidate genes with causative mutation potential using our Accu Tool. So we did something different to look at this. And that is, we went back to using a real phenotype, and we still have the 775 accessions, but only 40 of them had brown or red brown seed coats. So we went ahead and coded phenotypes and put the input into the Accu Tool. There were 19 brown and 11 red brown out of 775. We then asked for the modifying variants only from the genotype file and had the Accu Tool calculate accuracy, and the results we sorted by this pessimistic accuracy. And then we looked at the effects of the different genes. And the interesting or notable thing here is the abysmal pessimistic accuracy here, 3.7% for all of these. They actually have high average accuracy, but average accuracy does not take into account missing data, whereas this one does. And since there were 700 and some accessions with no data for phenotype, that resulted in a very low pessimistic accuracy. But nevertheless, this sorting feature highlighted this first variant here is a missense variant in this particular gene that causes an N to 1 at amino acid position 185 missense mutation. And it turns out that when we do inverse GWAS and we use that N185 missense mutation in this gene, and it turns out it's in an anthocyanin reductase gene, we can connect the inverse GWAS results. Remember, we sort by average accuracy when we're using inverse. So this is a synthetic phenotype on soy SNP 50K only. So we're returning those. So this marker is this marker in our original forward GWAS. So we connected these two, and that gives us some confidence, along with some other experiments that we did, to say that this gene, the ANR anthocyanin reductase 1 with this ID and this particular mutation, is the previously uncloned O locus gene. And so again, this is the phytosome view of this gene with its exons. This particular position is the causative mutation. It occurs in the third exon. And so this is a way accuracy can lead us to post-genomics or post-GWAS analysis to help us find genes and then use that information in our models or our parent choices to do a better job of crop improvement. So I didn't have time to go through the whole GWAS to gene strategy, and it's much more sophisticated than I laid out today, and it has many opportunities to add information to make your candidate gene list better. It turns out retrospective view is really quite simple. When you know the gene is cloned, it's easy to say, oh, look, there it is. When you don't know, it's much more difficult. So we have a lot of other ways to enhance our ability to help people find those genes. And we hope this is empowering so that other people, when they do a GWAS and they, instead of looking around for like, well, what genes are around here, they have some tools to enable them to identify a smaller list and hopefully the candidate gene and causative mutation itself. So in the end, I'm just trying to be a part of empowering other people to use genomics or genomic data so that we can design new genomes, new varieties with alleles that control phenotypes that are important to us. And to me, that's a successful applied genomics program. And I'd like to acknowledge that I didn't really come up with a whole lot of this. I just get to talk about it. I mentioned my work is really part of a two-group project. So I lead the biology group, which is mostly on the left, and Trupti Joshi leads the bioinformatics group, folks on the right. But the people really involved are Dr. Maria Shkrabasova at a university in the Czech Republic, and we've been doing Zoom for a while now. And then Nick Dietz, a PhD student, he really conceptualized and put into practice the Accu Tool. And Yin An Chan really converted it to something that could be available online. We've had some undergraduates participate in the work, another graduate student, and then several people on Trupti's team have been part of that data analysis. We've had funding from both the farmers, the university, and the USDA for that this work. And I hope my overall goal here was to inspire you to use a genomics thoughtfulness in your work and appreciate it maybe as we move on with new variety development. And so thank you for your time. And if there is time, I'd be glad to take questions.
Great. Thank you so much, Kristin. Okay, so I've known you for a while, right? Wait, let me turn this off.
Yeah.
So just you for a while, and I've been with you through a lot of the steps of developing the GWAS to genes. And I almost think that what's harder than developing the GWAS to genes is explaining the GWAS to genes. I like in your one list that you showed how you were able to go backwards from when you know the causative SNP mutation, but correlating that to the 50K marker set. So that way you can utilize the data that's provided in the GRIN, right? Sounds simple. But remember when we wrote that pod shatter paper and all the reviewers wrote back like, what are you talking about? So I really appreciate your presentation. I feel like it was coherent. It was very clear, and I understand the necessity of utilizing GWAS to leverage that data to go back and forth and see your data in multiple dimensions, not just, hey, here's the most associated SNP. So I did see a question from Phil McLean. Let's see. Phil McLean asks, have you applied this tool to quantitative traits?
So I had hoped that that first GWAS with the seed coat color would help you see that when multiple genes are involved, you can still do the trait. However, one limitation is when your phenotype is quantitative in the sense that you have a scale, you have 1 to 100, your values could fall between 1 and 100. What works really well in GWAS to genes is if you have ones or twos, like if you have two choices. So you have to kind of force a qualitative nature on a quantitative trait. So you have to have high or low or big or little, or the two choices really are what drives it. But if in a lot of quantitative traits where there are multiple genes, you can do that, because when you're looking at one location, the effect of that one location can often be compartmentalized into this bipartite, this two-part component.
Yeah, and I think a great example of that is maturity and flowering time. If someone were to ask me, Carrie, is maturity quantitative? I would begrudgingly say yes. But you and I both come from molecular biology backgrounds, and since those genes are also well characterized, yes, their combined effect influences maturity. But would you really consider it a quantitative genetics approach when you know the biochemistry and the molecular biology behind it?
Well, I'm a reductionist, so quantitative traits to me are combinations of qualitative traits that sometimes get masked and sometimes are unequally prevalent. I don't list them all as, it's just one. It's not black and white to me. I understand there's a lot of black and whites and sometimes they're gray. But that is my theory, that when you do a GWAS for a quantitative trait and you get 3 peaks, each one of those peaks holds a gene that you can find.
That's right. And then Phil McLean goes on to say, it seems like it might be time to abandon the 50K chip and then just spend money phenotyping your 775 lines.
Yeah, did you see the list? There were 111 glycine SOJAs. They vary for maturity group. There were 474 land races. No one location would ever want to produce even probably a subset of those. I do agree, though, for any other species, we think you probably need a minimum of 200 whole genome resequenced accessions that provide the diversity of phenotypes and genotypes, of course, for what you want to measure. We have had the shocking result that in our collection of 775, there was one accession that had a phenotype we were interested in. 774 had the, reference one alternate had the phenotype. We could clone the gene with that, with just one. So maybe you have a panel, and then if you have something unique, you get good sequence data, and we're working on a pipeline to make our panel expandable, but we're thinking maybe once or twice a year to expand it. But maybe what happens is you sequence your accession and then you utilize all of that other sequence that's already available.
Yeah, that goes along with the question that I was going to ask. I know we talk often about rare alleles, and how well do you think that these 775 lines can really capture, like how much genetic diversity do you think is present? And I know you said there's 100 SOJA lines, but when it comes to rare alleles, how accurate or how useful is this collection?
Yeah, for the very rarest alleles, it's not useful. Like you could ask the question, would I have found the hyoleic variants in these 775? I think the answer is no. I don't think there is a variant. Like we've done some other things with this data, which I didn't have time to present today. We have an allele catalog tool that allows you to look for any gene. What is the variation in that gene in this panel of accessions? And that's a really powerful tool as well. But for very rare alleles, you're basically dependent on in what genetic background did that allele arise. So if it was in a kind of a funky genetic background that's represented in the collection, then it will work to some degree. It won't point you to the exact causative mutation, but it can point you to the haplotype where it arose.
Okay. Yeah, I mean, that it makes sense. But you said that you are thinking of adding more, they just have to be resequenced, right? Like 50,
That's
I
right.
saw one line was 15x.
Yeah, we are targeting 15x right now, about $300 a pop.
Yeah, there you go. So soon enough, there'll be like 5,000 lines added to this collection.
There
right?
are 3,000 lines waiting for our pipeline to improve, but they are not GRIN accessions. So we're worried a little bit about bringing in genomic data for lines that we don't have much hope to get phenotype data for.
That makes sense. Okay. And then Andrew Skaboo from Missouri asks, how can you leverage this GWAS to genes approach to make use of the traditional mapping results that resulted in QTLs, which actually is, you could help clean up Soybase of how many like tens of thousand QTLs are on Soybase now. Let's clean that up and find the actual genes, right?
Right. So we have been working on some other avenues to leverage the whole genome sequence data to do that. As I mentioned, this allele catalog tool that I didn't have a chance to go through today, but it's also available on Soybase for anyone to use. And we are cataloging the variation in genes so that people can use that. For example, there's a gene that I have, a favorite gene, the raffinose synthase 2 gene, that I have a variant allele, but it's not a null. And in the allele catalog, I found a null allele in one glycine soja line, and I went in, obtained that glycine soja line, sequenced it, and it's there, like it's a true null allele. So we verified it. So I'll be using that to test the effect of that null allele and compare it to my other ones. So in genetic mapping studies, I think the usefulness will be questioning how rare is the donor of that in your mapping population, right? The parents that you chose, are they represented in the panel, or do you have to sequence them and put them in the panel? But you can certainly take your list of candidate genes, like Aaron was talking about, 17 candidate genes. He could look at the distribution of alleles for each of those 17 genes in the allele catalog that we have. And then whatever it is that is special about them, you could understand in those accessions.
All right. And then I don't know how useful this is, but you know, in some cases, you found that your actual candidate gene wasn't first or second, but maybe fourth or fifth down on the list. But have you tried maybe taking your most significant tagging SNP and then using the Accu Tool for the second or third most significant tagging SNP, and then comparing those lists? Has that helped to eliminate or get you down closer to the accurate gene?
Yeah, we did think about that strategy, and we do something like that in our actual more formal GWAS to gene strategy. So we have a series of ranking and filtering that involves actually doing another GWAS, GWAS 2, we call that, so that we include p-value. We haven't abandoned the statistics, even though I don't like it. We include the statistics and accuracy. We kind of decorate a p-value plot or a Manhattan plot with accuracy values to help identify what those best candidates are. So we have tried multiple avenues for that. And I feel like sort of empirically we've come, and this is work that Maria Skrabasova works on so hard, we've come to a pretty good pipeline, and we're anxious to get that full pipeline, which is a little complicated to understand, as you can imagine, out to the audience of users.
All right, Kristin, I got a stat question. Are you ready?
Okay, go.
Okay, this is from Nanoy Vandillo here at NDSU.
Oh, Nanoy.
Yeah.
Good work.
He asked, have you tried other GWAS methods in addition to MLM for qualitative traits, such as just a GLM, and what the accuracy would look like for the top significant SNP?
Yeah, if only I could have recorded all of the conversations Maria and I have had between MLM, a mixed linear model, and GLM, a general linear model for statistics-based methods, because I was fearful that MLM would suppress important things. I think accuracy is more like a GLM, right? It doesn't take into account these other population structure, kinship concerns. So we have looked at GLM. What happens with GLM is you get too many hits, basically. And so we do use MLM even in what we call GWAS 2, but we do think about that pretty hard. And accuracy doesn't involve that. So maybe we have an advantage there because we don't lose anything if we calculate accuracy.
Oh, so your Accu Tool isn't using an MLM or GLM model?
No, accuracy is mathematically that like 100%, 0%. Like it is a direct correspondence calculation. Average accuracy ignores missing data. Pessimistic accuracy takes into account both the skew of wild type and mutant frequency, and it assumes every bit of missing data is wrong.
Okay. Oh, and then Aaron Lorenz is asking, is the Accu Tool publicly available?
Yes, so just navigate over to soykb.org, and then there should be a tab of different things you can click on, like a menu sort of across the top, and there's one that's tools, and Accu Tool is down halfway down the tools tab. And not to be confused, because I did talk about today another tool on that same tab of tools is Soybean Allele Catalog, which is a lie. It's a different tool. But if you go under Browse, the Soybean Allele Catalog is the other thing I was talking about.
Oh, you're going to have to come out with a, what is it called? A white sheet for how to navigate your website. Because
we've really struggled with how to explain some of this.
Yeah, I mean, because there are more useful tools on that website, like your SNPviz. I know it took me a while to get it, but your SNPviz tool is also hugely useful. And just briefly, SNPviz is a way to put in an interval on a certain chromosome, and you can see all the variations from whatever whole sequence lines that you select in the beginning, and you can see all the variants in this interval, which I was developing markers and I needed to know variants that were in, this 200 base pair range. And I literally had no idea how to do that except for using SNPviz. So the
SNPviz is the part of the formal GWAS to genes pipeline that would take, I don't know, we will have to have a whole webinar series on GWAS to genes. I think Marie and I have talked about that workshop.
Oh, and then Phil McLean asks, what do you think about using Lee as a reference?
Oh, so I'm of the opinion, and Maria and I agree on this, that we would like to create the gold standard gene list for all soybean genes. And so I'm not so excited about Lee. I'm excited about the glycine soja reference genomes. And there are multiple ones. We have so many reference genomes now. This is obviously something that's important for common bean as well, with the two, whatever you call those races of common bean. The genome that you use as your reference genome is really an interesting and intriguing aspect. And right now we don't have a project to remap. For example, I'd like to remap all of the glycine soja sequences against a glycine soja reference sequence. So I'm excited about having more data available. And frankly, we're behind in the tools. Like, we can't keep up with the data generation and analysis speed to make our applied work appropriate to the data that's available. Like, my team is pretty small right now, our team, and we would like to get some more funding to pursue some of these things. Like, now it's cheaper to create the data than to analyze it, for sure.
All right, fantastic. I don't think that there are any more questions. And I know this conversation is nice because you can see it comes up with a lot of like genomics philosophy. And so this conversation could go on for hours. So next year when we're in person, because now we would be going to the reception and to dinner, we can save it for next year. So I want to say thank you again to Kristin Bilyeu.
Thank you so much.